I routinely explain science to non-scientists. And so when I've been explaining one of the studies that we do with epigenetics, what I say is that there's genes that make proteins, and those genes have to be controlled. And so epigenetics is one layer of controlling which proteins get expressed. Environmental factors and biochemistry and sort of all the chemicals in our environment, and water is a chemical, and the molecules in the food we eat all impact our biochemistry. We are a walking bag of biochemistry. Epigenetics is just one little piece of that biochemistry. And so it's, although we think it's tightly controlled, there's no inherent reason why epigenetics cannot also be modulated by the biochemistry of our environment that becomes part of our internal environment, which is just another way of saying it's just part of our body, and our body responds. The bodies of any living things respond to their environment, so we should expect it to. We don't really know yet, and that's one of the fun things. That's what makes not only science fun, but also this particular field fun, is that it's relatively new, and since science is all about finding out new stuff, this is a fun place for a scientist to work, because we really are finding out new stuff about one of the most critical pieces of life, which is genetic control. We'll know what the big changes are and how to avoid those, or what to do to avoid those. And we'll know what changes are trivial or trivially small, and that they happen, but they have no consequence. And so we don't have to worry about them. And in fact, that's the real challenge of risk assessment, is separating the big effects, the effects you have to worry about, from the effects you don't have to worry about. Hybrid vigor is a real thing, and hybrid vigor in science, or cross-pollination, is a wonderful thing. And getting scientists together with different backgrounds and different points of view, because they're scientists, they're coming in with a perception of, okay, I don't know very much. I know a little bit about this thing, but in the broader scheme of things, I don't know very much. You know about stuff that I don't know about, so let me learn from you. So what we've got here is a whole room full of people who are eager to learn from each other, and all of whom can chip in a little something into the pot, the larger pot of knowledge. And so we all become smarter. And if we're lucky, then we'll also get a good idea about what the next set of experiments ought to be, or what's the shortest way to gain the most useful knowledge, not the greatest amount of knowledge, but the most useful knowledge. Epigenetics, in the next 10 or 15 years, we will, I hope, if it's like other fields, we will be at the point where we know what things are important and what things are just sort of the background biological noise, and therefore we'll know which things to really focus on and which ones are sideshows. And in fact, all the big areas of biology have gone through that kind of transition. When I was in graduate school 40 years ago, the whole gene, RNA, protein thing was the hot area, and that's where people were working on. And now that's just a snooze. That puts people to sleep in high school because it's so boring. And so epigenetics will be in the spotlight for a while. A lot of information will be generated. We'll figure out what of that information is really the most important, and then we'll move on to applying that into the broader scheme of risk assessment.